So good afternoon from me as well. My name is Lorena Skevi, and I'm coming from University of Bath, where I'm currently doing my PhD. So today I'm presenting our recent work on later age cracks in concrete using bacteria. And by later age cracks here for this study, I'm referring to cracks created in 9-month-old mortar samples. So this investigation comes from the fact that so far self-healing with bacteria has shown promising results for cracks created in concrete at earlier ages, usually 7 or 28 days. However, cracks may occur throughout the lifetime of the structure. So the question is whether this technology still works when cracks appear in older concrete. So to understand the mechanism behind self-healing concrete, it helps to take a look on natural self-healing behavior, because it is a technology inspired by nature, such as our body and our bones shown here. So in general, the key principle of the mechanism lies in the release of healing agents in the fractured area. Immediately after the detection of the fracture, chemical signals are released, and these agents are moved to the fractured area, which then start the healing. So in a similar manner, in self-healing concrete, we have healing agents that are embedded in concrete. Here, so in our case, we incorporate bacteria and we encapsulate them in aerated concrete granules. These are shown here in a green color. And also we add growth medium, which are necessary for the growth of the bacteria. And here they can be barely seen as yellow dots. So when a crack occurs, it propagates through these aggregates, since they are the weakest part of the mortar. So the spores are then released, and they come in contact with the growth media, as well as oxygen and water that flows through the crack. And under these conditions, the spores can germinate into vegetative cells, and then through their metabolic activities, they can precipitate calcite and seal the crack in this way. Our bacteria that we are using, the Bacillus coni, they do that by oxidizing organic source. So they need to have an organic source added in the growth media. And by respiration, the byproducts of this process is calcite and carbon dioxide, which carbon dioxide will further react with calcium hydroxide that is present in concrete, resulting in more precipitation of calcite. And also the cells of the cell wall of the bacteria is often negatively charged, and they can also act in that way as nucleation points, so that calcite crystals form around them. Now, for applying this mechanism, we have developed a methodology over the past years in Bath that we used in this study as well, and which I'm going to briefly describe. So as I said, we use the ACGs for encapsulating the bacteria. What we do is that we impregnate the granules in a water solution, where we add the spores. So we do that under vacuum, so that we achieve a better absorbance. And this is how the ACGs look at the end of the process. And we make the biosamples, so the spores that contain growth, excuse me, the samples that contain spores and growth media, by replacing 6% of the sand with the spore-bearing ACGs. We add the growth media, which comprises of yeast extract and calcium nitrate in the mixing water. And here is the mixed designs for the samples that we prepare. So apart from biosamples, as you can see, we have two more types which are used as benchmarks. One is a plain mortar sample, basically the reference, and the control one that contains only growth media, in order to assess the effect of the growth media on the healing. Then we crack the samples in 3-point bending load in order to control the opening crack and avoid a complete split. The samples are wrapped with the carbon fiber reinforced polymer strips. And also the crack width is monitored by a crack mouth opening displacement gauge, so that we have a single crack of 0.4 to 0.5 millimeter width. So one set of samples, we cracked them after 28 days of curing in water, while the other set of samples. After this time, they were moved to a conditioning room of stable temperature and relative humidity, where they were stored for 9 months until the day of cracking. And finally, for the healing condition, after cracking, the specimens were put in separate containers by sample type, and to avoid any leak of growth media or spores. And they were semi-submerged in water. So in order to examine here the effect of the temperature on the healing, one set was stored at room temperature at approximately 20 degrees, and the other one were stored in the fridge where the temperature was set at 7.5 degrees Celsius. So we assessed the healing in one way that we assessed the healing was by optical microscopy, where the crack widths were measured immediately after cracking, and then weekly until 56 days of healing. And the percentage was calculated by equation 1. And we also performed the water flow test to assess the recovery of water tightness as the samples were healing. And the setting of the test can be seen in the photo here. The water flow coefficient was taken by equation 3, and the geometric parameters of the equation are depicted in the photo. What we measured during the test was basically the time that water needed to flow from a standard distance, so from H1 to H2. And we did that test after 28 and after 56 days of healing. So we got the healing ratio here in terms of water tightness. So here we see the results in terms of crack closure as they were measured by the microscope. So in each of the graphs, we see 3 lines for each type of the samples. So the reference, the control, and the biosamples. Now, the one first remark that we can make just by one look at all the graphs is that a significant percentage of healing is achieved at the first days of the healing, the first 7 days of the healing. So we also see that the reference samples, which is a plain mortar, present more or less the same healing for both ages and at both temperatures. And then the control and biosamples present some differences. So we see that for 28 days old samples, the effect of the growth media, so here seen as the control, the blue line, is significant, which is not the case for 9-month-old samples, where we see that growth media on their own do not contribute as much to the healing, but at this age is the biosamples that contain the spores that present significant healing. So that could be happening for a couple of reasons. One of the main one, more actually, is that calcium nitrate that we added in the growth media is, as we said, a calcium source. So what happens at 28 days old samples is that when a crack occurs, atmospheric carbon dioxide reacts with this additional calcium, resulting in the formation of calcite, carbonation reaction, basically, that contributes to the healing. On the other hand, in 9-month-old mortar samples that are probably already carbonated before the crack appears. So when the crack takes place, there is not so much calcium in the material available to react with carbon dioxide, and therefore the contribution to the healing from the controls is insignificant. And what also happens in these carbonated samples, on the other hand, is that the pH drops, and that makes the environment in concrete more friendly for the bacteria. And therefore, we see that the spores contemplate for the reduced healing effect of the growth media. And finally, we should note that the healing of 28-day samples with bacteria seems to be impaired at 7.5 degrees, since it's less than the control. And this is probably related to the type of the bacteria we added, because the B. coni that we added in this experiment is a mesophile bacteria that grows best at 20 to 30 degrees. So it seems that here the lower temperature in combination with a higher pH, did not allow the bacteria to perform a sufficient healing. Now, similar results were obtained by the water flow test as well. So we see that the biosamples are performing better for 9-month-old mortar samples, while for 20 days old mortar samples, the controls are also performing some healing. We should also mention here that even if the healing ratio in terms of crack closure was observed to be around 60 to 70% for biosamples, here we see that the water tightness is higher, so it is more significant after 56 days of healing, that is. And this is a visual observation of what I described. We see that here that 28 days old samples, both control and biosamples show some healing in comparison to the reference that shows none. And on the other hand, on 9-month-old mortar samples, only the biosamples show healing, and that is at 20 degrees, while at 7.5, we see that the control here show bigger, larger, not larger, but higher crack closure than the biosamples. Oh, and the opposite is observed at 9 months mortar samples. So as a conclusion, and to answer the question that we asked in the beginning, so it seems that this technology seems to be efficient for later age cracks in concrete, here created after 9 months. It is important because it means that the spores are well and happy, and it is a relief to know that they can still wake up and promote the healing whenever the crack occurs. And also, we see that the contribution of the growth media to the healing is reduced for cracks at older concrete. And finally, we saw that the temperature is a factor that should be taken into consideration, especially for earlier cracks, for newer samples. Overall, the bigger scope of this technology is that it wants to be a step towards more resilient structures and achieve the goal for less CO2 emissions that has been set for the construction industry these years and for the years to come. So I want to thank you all for your attention, and I'll be very happy to answer any questions if there are. Thank you.